Hi friends, myself Anaga Srinivasan, first year MSc Biology student. Here my topic is immunotherapy. Let's see more about immunotherapy. First of all, we all want to know about what is cancer. We already know that cancer is the uncontrolled proliferation of abnormal cells in the body. The cancer treatment is dependent on the type of cancer, if its stage is cancer has spread or not, and the general health. The goal of treatment is to kill as many cancerous cells while minimizing damage to normal cells nearby. There are so many treatments for cancer. Let's see what are they. Surgery. Surgery is the directly removing of the tumor. Chemotherapy is a therapy that done with the help of chemicals to kill cancer cells. Radiation is the usage of X-rays to kill cancerous cells. Immunotherapy is a therapy which boosts immune system against cancer cells. Nowadays, most of the cancer patients are treated through chemotherapy, but immunotherapy is better than other treatments. Because we all know chemotherapy is a therapy using chemicals to kill fast growing cells, including both cancerous and non cancerous in the body. While immunotherapy helps the immune system do a better job of identifying cancer cells so it can attack and kill them. Let's see much more about immunotherapy. So, what is immunotherapy? It is a treatment that uses certain parts of the immune system to fight diseases such as cancer, stimulating your own immune system to work harder or smarter to attack cancer cells, giving you immune system proteins such as man made immune system proteins. Dr. William Coley, who is a neurophysician, used Coley's toxins in the late 80s as crude immunotherapy with some success. Coley tried to develop bacterial extract that can be injected under skin and stimulate patients' immune system to attack and destroy malignancy. Coley's results confirmed that body has capability to destroy tumor even after it spread. There are three groups of anti-cancer strategies. Those are that depends on antibodies or immune cells to attack tumor cells. Next one is those that inhibit activity of cancer promoting cells. And the last one is those that prevent the growth of blood vessels that nourishes the tumor. In recent years, two broad treatment strategies involving immune system have been pursued. The first one is passive immunotherapy and the second one is active immunotherapy. Passive immunotherapy. Treat cancer patients by administering antibodies as therapeutic agents. These antibodies recognize and bind to specific proteins on surface of tumor cells being targeted. Once bound, the antibody orchestrates an attack on the cell carried out by other elements of immune system. Investigators produced humanized antibodies that are largely human proteins except for relatively small parts that recognize the antigen which remains mousy. In past few years, researchers produces antibodies that have completely human amino acid sequence. In one approach, mice have been genetically engineered so that their immune system produces fully human antibody molecules. Monoclonal antibodies approved to treat cancer and other medical conditions. Here are the examples for humanized antibodies. The first one is Herceptin. Herceptin is a humanized antibody directed against cell surface receptor HER2 that binds a growth factor that stimulates the proliferation of breast cancer cells. Herceptin inhibits activation of receptor by growth factor and stimulates receptor internalization. Approximately 25% of breast cancers are composed of cells that overexpress HER2 gene, which causes these cells to be especially sensitive to growth factor stimulation. Clinical trials demonstrated that Herceptin is effective either alone or in combination with chemotherapy in slowing the growth of breast cancer in significant fraction of patients with HER2 overexpressing tumors. A 2005 study of 3,000 Women with early stage breast cancer reported that Herceptin reduced chance of reoccurrence of disease by about 50% in 4-year period. The most effective humanized antibody is rituximab, which is approved in 1997 for the treatment of non-Hodgkin's B-cell lymphoma. Rituximab binds to cell surface protein CD20 that is present on malignant B cells in approximately 95% of cases of this disease. Binding of antibody to CD20 protein inhibits cell growth and induces the cells to undergo apoptosis. Introduction of this antibody has completely reversed the prospects for people with this particular cancer. People that would have once had a very grim prognosis now stand an excellent chance for complete remission of their disease. Next one is the fully humanized antibody panitumumab directed against the EGF receptor has shown considerable promise in treatment of metastatic colon cancer. Because it is a human protein, panitumumab remains in circulation long enough to be administered once every other week. A new generation of antibodies developed containing radioactive atom or toxic compound conjugated to antibody molecule. Antibody targets complex to cancer cell and associated atom or compound kills targeted cell. The two radioactively labeled anti-cancer CD20 antibodies are Sevalin and Betsar have been approved for treatment of non-Hodgkin's B-cell lymphoma and toxic drug-linked antibody Mylotarg has been approved to treat acute myeloid leukemia. Next one is the second method of immunotherapy, which is active or adaptive immunotherapy. It is the approach that tries to get a person's own immune system more involved in fight against malignant cells. The immune system has evolved to recognize and destroy foreign materials, but cancers are derived from person's own cells. Although many tumor cells do contain proteins that are not expressed by their normal counterparts or mutated proteins, example, RAS. RAS that are different from those present in normal cells, they are still basically host proteins present in normal cells. They are still basically host proteins present in host cells. As a result, the immune system typically fails to recognize these proteins as inappropriate. Even if a person does possess immune cells, that is, T cells that recognize certain tumor associated antigen, tumors evolve mechanisms that allow them to escape immune destruction. Many different strategies have been formulated to overcome these hurdles and artificially stimulate the immune system to mount a more vigorous response against tumor cells. Immune cells are isolated from patients, stimulated in one way or another in vitro, allowed to proliferate in culture, and then reintroduced into patient. In some studies, isolated immune cells are genetically modified before proliferation to enhance their tumor attacking potential. A significant minority of patients have exhibited a positive response to treatment, which means that their tumors have at least shrunk in size or extent, and their expected time of survival has increased significantly. Next one is Provenge, which has shown promise in clinical trials in patients with advanced prostate cancer. It is the first of these treatments likely to be approved. Provenge is designed to mount an immune response against cells containing a particular enzyme, prostatic acid phosphatase, that is expressed by this cancer. The ultimate goal of cancer researchers is to produce immunopreventive treatments in which people would be vaccinated with antigen that would prevent them from ever developing life-threatening cancer. Here is a reference. Thank you all for listening my class.